Secondary active transport. So this is a type of transport that does not directly use ATP, but it uses ATP indirectly or secondarily. And it's pretty important. So if you remember, primary active transport is what's shown over here. The sodium-potassium pump is going to be our primary example of primary active transport, directly breaking down ATP in order to do something that is not spontaneous, to move ions against their concentration gradients. I'm actually going to add here, this is high potassium inside the cell. This is high sodium outside of the cell. So when this protein is moving sodium out and potassium in, it is helping to maintain that difference. We'll get into the details of why that needs to be maintained, various reasons. There's leak, and then there's processes that happen that need this to be constantly maintained. So that's that primary active transport. Secondary active transport, this protein itself does not use ATP, but it cannot function without the gradient that was set up over here. So in this case, glucose is what we're actually looking at transporting. So glucose is low outside of the cell and high inside the cell. But we want to get glucose in the cell still. This has applications for urinary, digestive system. How can we do that? We can't just use the glucose carrier molecule. First of all, we do need a protein, right? Glucose can't pass across the phospholipid bilayer. It needs a protein. Usually it uses a carrier. It often does use just a facilitated diffusion carrier. It can't in this case because there's no drive for it. We need something different. So it's a different molecule here, this different protein that carries glucose in this case. So we want to be able to move glucose in. We're going to use co-transport. Both sodium and glucose are moving in together. That is called co-transport. This is spontaneous. Why? Because there's high sodium outside. High sodium outside, low sodium inside creates a drive for sodium, right? The drive for sodium is this way. It's going to pull glucose with it. So this sodium-potassium pump is kind of like a battery, right? The stored energy of high sodium outside, low potassium inside is allowing for other processes to work. Co-transport is one of those processes. The action potential is going to be another one. So ATP is still used, but indirectly. It's used to maintain the sodium drive. And I wanted to preview something that reason is going to be important right here. So this is kind of more for those of you who like some application. This is an example of when this would occur either in the kidney or the intestine. So what's happening here is there's low glucose inside the lumen of the kidney or the intestine. Glucose is high inside the epithelial cell and then low in the extracellular fluid. It's going to be carried away into the bloodstream after this. What needs to happen is glucose and sodium need to be able to move inside the cell together. Glucose can't move in without sodium's help. So that's step one here, that co-transport using that sodium gradient. Glucose can then here facilitate diffusion. That's just the glucose carrier, because here we're going from high to low. This whole process can't be maintained, can't keep going without this. This is our primary active transport, our sodium-potassium pump. Again, sodium-potassium pump is one example of an active transport, is the most common example that I will talk about. So over here, we've got the sodium-potassium pump that's constantly maintaining that low sodium inside and high sodium outside that's needed for this to happen. So super cool example of why this matters. I would want you to be able to draw out like these three proteins to demonstrate you understand primary active transport, secondary active transport, and facilitated diffusion.